Hello, this is Eva Amson welcoming you to this Bite-Size Bio web seminar, which today is sponsored by Refine. Refine are pioneers in mass photometry, a revolutionary new way to analyze molecules. Mass photometry enables the accurate mass measurement of single molecules in solution in their native state and without the need for labels. This approach opens up new possibilities for bioanalytics and research into the functions of biomolecules. Today's presentation is titled Applications of Mass Photometry and is presented by Justin Benesch, who is Professor of Physical Chemistry at the University of Oxford, as well as Chief Innovation Officer at Refine. And he is joined by Nikolas Hundt of Ludwig-Maximilians-Universität in Munich. As always, we will have a question and answer session after the presentation, so please type any questions you have into the questions box, which appears on the right-hand side of your screen, and I will put them to our speakers at the end of the talk. The recording of this webinar will also be available on the Bite-Size Bio website. So now, over to you, Justin, for the presentation.
Hi, I'm Justin Benesch. It's my real pleasure to talk to you today about what I've titled Weighing Heterogeneous Protein Assemblies, by which I mean actually measuring the molecular mass of proteins and protein assemblies and how they interact with each other and actually other components of the cell. And I'd like to sort of start off just by sort of throwing the idea out that when we think about weighing things, we obviously think about measuring their mass. That's the sort of the fundamental observable that we get from a weighing experiment. And what I'm going to try and convince you today is that we can actually get information from that type of experiment about the structure of molecules and protein assemblies and specifically and about the thermodynamics that control that structure, that represent the interface and the interactions within it. And we can get information about the kinetics, in other words, the rate constants that underpin those thermodynamics that I just talked about. So in the example here, on the right-hand side, you have a protein assembly. It's made up actually of 12 identical subunits. And you can kind of see it looks a little bit vaguely sort of triangular, sort of a triangular disc. On the top face, there are 3 dimers, and there's the same on the bottom. You can't see them, obviously. And you'll see, so there's a blue dimer, and there's sort of light greenish blue dimer here, and an orange dimer there. So already we have different elements of structure just within an organization such as this. And it goes beyond just thinking about where individual atoms are, but we can break it down into the fact that this is, as I said, a triangular disk of which there are 2 on top of each other, that each disk is made of 3 dimers, each dimer is obviously made of 2 monomers, and overall the shape of the molecule looks vaguely toroidal. So I'm going to try and illustrate how just by measuring mass and measuring those subcomponents of this oligomer that I just talked about, that we can actually sort of reconstitute the structure to a certain extent, understand ultimately what governs the assembly of these proteins. And then, as I said, I'm going to try and do that in a quantitative way, so talk about the biophysics that underpin that structure. So the 2 methods I'm going to talk about, as you probably anticipate, will be mass photometry, so that's indicated here on the right. So mass photometry, as I'll talk a little bit more about in a second, is a technology that weighs mass by leveraging the interaction that molecules have with light. And I'm going to complement that with talking about native mass spectrometry, which is on the left-hand side, which uses a different type of physics, namely the way molecules, specifically in their charge form, so ions, how ions move in vacuum under the influence of, in general, electric fields. And I'm going to finish off the talk then with one sort of quite detailed example where I'm actually going to use both of these 2 methods in a way that we can get more information now about the system we're looking at than if we'd used either of the 2 in isolation. So let's start a little bit and talk about mass photometry. So I'm sure most of you are aware a little bit about what mass spectrometry is, otherwise you wouldn't be listening today. But I'm going to try and go through the principles in a sort of a simplistic, from a simplistic standpoint. Ultimately, the methodology works by having a droplet of solution, a small quantity of solution, on top of a glass slide. And so that's represented here. And in that droplet of solution, we have our analytes of interest, right? So I've just color-coded them here in 7 different colors for the 7 different molecules. And you can see, broadly speaking, 4 of them seem to be around about the same size, and a couple, or 5 seem around the same size, and a couple seem smaller. And they're sort of in solution at varying distances away from the glass slides, the surface that we're actually going to use essentially as our detector. So that glass slide is illuminated, so there's incident light on that glass slide. Some of that light is reflected, and for the molecules that are close to the surface, such as this yellow one here, those molecules will scatter some light as well. So you have scattered light, and that scattered light can actually interfere with the light that's reflected, and it's this interference that we actually measure in the mass photometry experiment. So what does this look like? Well, schematically, we take an image on our detector on the camera within the instrument, and the molecule that was near the surface, basically touching the surface, gave a significant amount of signal, right? So this is our interferometric contrast. And then what we do is, rather than just recording one image, we record a movie. So we get multiple video frames, and we video in such a way that we're always looking for differences that have occurred between the frames before the preceding frames. So what you see is molecules that weren't visible in the first frame. Let's say this dark green guy here was too far away from the surface to be detected in the initial frame, but starts to appear over time, ultimately giving rise to a strong contrast in this 5th frame here. Whereas the one that already gave you significant signal in the first frame, because that no longer changes, that essentially starts to, the signal that from that starts to decay, and in the 5th frame you no longer see it. So we get a sort of a time-resolved measurement of molecules as they approach and contact the detection surface, which is named the cover slide onto which the droplet has been put. So if we measure and record the contrast in each of these frames, in other words, contrast over time for each of the individual molecules we detected, so they're all color-coded, what happens is that sometimes, or if we follow some of them individually, this orange guy, that signal just decays with time, so time goes down from top to bottom here, whereas some, the contrast increases and then decreases. And what we really care about is what the maximum contrast is that any single molecular event returns, right? So this blue molecule here gives us maximum contrast in the 2nd frame and then decays. So we can detect and measure that maximum contrast for all of the molecular events that we see, and then we just sum up or bin those maximum contrasts. So in this case, we had 5 molecules that appeared in this contrast window, 1 molecule that had about 3/4 of that contrast, another one that had about half of that contrast. So that's the basic principle of how mass photometry works. So let's just take a look at some data. And so this is a video, so those frames I just showed you before, and you can see lots of dark spots. They appear and disappear. And each of those corresponds to a molecular landing event. In this case, just for the most boring of all proteins, BSA, right? And so some of the BSA molecules are monomeric, give a certain amount of signal. Some are dimeric or even larger, and proportionally give larger interferometric signal. So we can sort of look at this video and pick out molecular events, and I can show you 4 characteristic maximum contrast images that we can ascribe to 4 individual oligomeric states. So the monomer, the dimer, the trimer, and the tetramer. And you can see that those events, those dots, vary in their darkness, right? So clearly the one on the right has got much higher contrast than the one on the left. So that is essentially giving us the impression that we're not just detecting molecules, but we're detecting them actually in a quantitative manner. And to sort of interrogate that further, the way you can view your data is to think, well, okay, we have contrast that we measure for each molecular event, and we're measuring molecular events as a function of time. And so if you look at that, you see the scatter plot. Most of our events for this particular movie of BSA had a contrast value which is just under 0.5%, whereas there are some events that have double that, triple that, quadruple that, and that's very much consistent with the idea that we might have multiple sequential oligomeric states of that particular protein. So then on the top here then is the experimental histogram that we had for BSA, and you can see that though you're mainly monomeric, there are significant proportions of dimers, and if you zoom in closely, you can see some trimers and even some tetramers, even though the tetramers themselves correspond to less than 1% of the overall abundance in solution. So this essentially then is a mass measurement, and I can prove that to you by this graph that we published when we first developed this methodology, which shows that if you compare the sequence mass, in other words, the mass that you expect for a protein of interest, with the contrast that you measure, there's a very straight line, and the error, so in other words, the deviation from that correlation, is on the order of just a couple of percent. And particularly at higher masses, that really diminishes down to a very small error comparable to other methodologies, or compared to other methodologies out there. So that's sort of the background to the methodology. And what I'd like to talk about today is actually using mass photometry on its own and in concert with mass spectrometry to look at heterogeneous molecules. So what I've got here then is a video of a protein that we've been working on in my research group for over a decade now. And if you watch the movie, you will see there are dots that are dark and there are dots that are darker. In fact, if you watch it, you will see what appears to be really quite a broad range of contrasts in that video. And so if we plot that data, that histogram, you can see on the x-axis, this is the contrast. We can convert that by calibration to a mass axis on the upper x-axis. And we can clearly see a range of oligomeric states, things that we can ascribe to the dimer of the protein. We know its monomeric mass is 22.5 kilodaltons, but then tetramer, hexamer, 8, 10, 12, 14, 16, etc. Right, so this protein, just by measuring its mass distribution, we already know a few important things. So firstly, the protein is clearly oligomeric, right? It forms a number of oligomeric states. It's polydisperse because it doesn't form one oligomeric state. It forms a range of them at equilibrium. And we know that the protein seems to be composed of dimeric subunits because we see only oligomers that have an even number of subunits, like the 16-mer and the 18-mer here. So even just in this case, just from measuring the mass distribution, we already have significant structural insight into the protein. So this is the human form of the protein, HSP27. And I want to show you that if we compare that protein measured by mass photometry, which is now the gray histogram in the background, with our most state-of-the-art, most complicated native mass spectrometry experiment, which I'm going to talk in more details to how we do that experiment later on, but you'll see the mass distributions align really quite well. And this is a huge boon because the native mass spectrometry experiment by its very nature is a gas phase vacuum-based experiment, which I have to say is not the easy experiment to do, whereas the mass photometry experiment really is a matter of 1 or 2 minutes worth of experiments, and crucially is all done in solution. So these are sort of mutually validating experiments in my view. On the left-hand side, conversely, is a monodisperse protein. Of course, there's some disassembled parts here, but essentially this protein is focused in one oligomeric state. So very quickly, you can compare a monodisperse and a polydisperse case by doing the mass photometry experiment in a way that we've sort of independently validated here using mass spectrometry as a background validation tool. So given the advantage then of using mass photometry in terms of measuring solution phase mass distributions and doing so in just a minute or two, it lends itself very well to screening. And I've just given two examples here. The first is taking the same protein I just showed you before and screening it under conditions of manipulations in order to try and force it towards monodispersity. And you can see if you look from the left-hand side to the right-hand side, clearly the mass distribution narrows and becomes more confined ultimately to a single oligomeric state. So in the realm of structural biology, and we're interested ourselves in trying to characterize the structure at high resolution, this type of polydispersity is of course a major headache, whether it's for crystallography or NMR or electron microscopy. The fact that you have such a large ensemble of oligomeric states coexisting at equilibrium means there are significant hurdles towards structural characterization, whereas here we can screen approaches that we've taken to try and reduce that dispersity, such that getting high resolution structural information for this form on the right hand side seems a realistic possibility. On the left hand side on the bottom column, bottom row, I just wanted to give one example with the protein that is also polydisperse. Again, covers a wide mass range. In this case, there is no resolution between oligomeric states like there is here, because the mass of the cell unit is so small. But we can actually change conditions so that we can watch the protein shifting from an average mass, which is near 700 kilodaltons, to lower, and ultimately ending up somewhere below 500. So this again gives us information about the assembly and the dynamic oligomerization of this molecule. So I've told you a little bit about mass photometry and how it can be used, and I'd like to introduce briefly now an alternative mass measurement approach, this mass spectrometry, just so to give some context for when we're going to combine the two methodologies in the last few minutes of this presentation. So mass spectrometry works on taking molecules in solution and putting them into the vacuum of a mass spectrometer. And the idea behind native mass spectrometry is that you do so in a way that the solution state structure is preserved for the duration of the mass spectrometry experiment. And I'll just show you how the experiment returns data. So this is the same protein I showed you before, made of 12 subunits that are identical in mass, each just about 16, nearing 17 kilodaltons each. And what you get is a series of charge states, a series of peaks that are separated on a mass to charge axis, not because they differ in mass, but because they differ in Z, so the charge that they carry within that mass spectrometer. So this is the mass spectrum then for the monodispersed 200 kilodalton oligomer, whereas in a non-native mass spectrum, you would expect to have seen monomers down at low mass to charge. Now, what we can do, and bear with me please, because I'd like you to be able to understand how we do the experiment so that the data that comes doesn't seem hard to follow. But what we can do is we can select a particular charge state, such as this one here, and then we can submit it to activating collisions in the gas phase. And the only take-home message I want you to get from this is that a protein that started off with a certain number of subunits, in this case 12, as we increase the activation energy, loses successive monomers to become an 11mer, so having lost one subunit, and then to become a 10mer. And if you look at the separation between peaks here and here and here, it's clearly much better having removed a couple of monomers. And that's because the monomers carry away quite a large amount of charge with them. So on the M over Z axis, you're then relatively decharged and your peaks are well separated. So if you do that instead for a protein that's not monodisperse but is polydisperse, the value comes that increased separation between peaks at higher activation energies and oligomers that have lost a number of subunits is such that you can actually resolve all the oligomeric states that underlie there. And you can use that resolution to quantify the oligomeric states that you have. So that's how we do the experiment. So this is the result. What I've overlaid here are 4 different ways of measuring mass of heterogeneous protein assemblies. So in the background in black is a sort of a crude estimation of a mass distribution obtained from negative stain electron microscopy. In the red line is size exclusion chromatography coupled to multi-angle light scattering. In blue is the native mass spectrometry experiment that I just described to you, and in gray is mass photometry. I think the first thing to note is that they all correspond to each other pretty well, which again is mutually sort of validating. There are some sort of biases that we might imagine in each case, and I'm not going to spend too much time talking about them. But both the native mass spec and the mass photometry are sort of instant detection methodologies, as I would probably say is the negative stain electron microscopy in this case. In other words, there are no mass transport effects that might, for instance, confuse sec-mols data. The native mass spec is very high resolution, but is limited by the fact that we know we're measuring a distribution in vacuum, and we're beholden to the manipulations that I just described in terms of activation and dissociation to obtain that distribution. And mass photometry gives ultimately the a bona fide solution phase distribution that matches very well the other methodologies, and is slightly shifted to higher mass than the native mass spectrometry, and that's actually, I would say, due to the fact that native mass spectrometry will bias to slightly lower masses given the collisional activation process I just described to you. And I'd like to, if you bear that point in mind, we'll come back to that in the last part of the talk. So I'd like to finish then with a few minutes describing, a biological problem that we've actually been tackling for a number of years, and we've been interested in a series of proteins that are called the small heat shock proteins. They're an important part of the cell's tolerance and response to stress. That might be environmental stress, or it might be stresses such as aging that lead to various disease states. And the proteins are not just important, they've also turned out to be really quite challenging to study by a whole bunch of methodologies. And I'm just going to focus on one quadrant of this chart. I'm going to talk about the fact that these proteins have a real tendency to oligomerize, and not just oligomerize with themselves, self-assembly, but also to co-assemble. In other words, forming mixtures or oligomers that are mixtures of subunits, heterooligomers. And given that the proteins are oligomeric, and there's several, well, in humans there are 10 small heat shock protein genes. In other organisms there are often many more than that. Given that complexity, what can we say about the possible ensemble of states that these proteins populate? So the example I'm going to give you is 2 small heat shock proteins from humans, HSP-B1 and B5. They're probably the 2 most well-known human small heat shock proteins, and they're found together in a range of tissues, but probably most importantly in muscle tissue. And we can obtain, through the experiments I described before, the native mass spectrometry experiments, we can obtain very precise oligomeric distributions for the proteins on their own, right? So take the red one is HSP-B1 and the blue one is HSP-B5. And if you take those 2 proteins and you mix them together, you obtain a mass spectrum, this one here at the bottom, which is clearly not just the superposition of the 2 that are above, right? There are the peaks that stick out, for instance, this one and this one, in the top 2 mass spectra do not appear similarly in the bottom spectrum. So these 2 proteins are clearly interacting, and the only way that would be feasible really is whether they form heteroligomers such as this. And so the challenge really now is one of quantifying that heteroligomerization and understanding the biophysical basis for it. So what we do is we do a tandem mass spectrometry experiment, as I described to you before, in which we interrogate just a specific part of the native mass spectrum, right? So we can select a certain population of ions, transmit them into the mass spectrometer, or transmit them through the mass spectrometer into a collision cell, whereupon we activate and dissociate the molecules, and then look at the fragments in the way I described. And if we do that for this particular slice, we get a range of different species that we can assign by deconvolving that mass spectrum into a range of different oligomeric states, which are shown here. So what you see in this case, for that particular isolation, we saw oligomers made of 15, 16, 17, 18, 19, 20 subunits, but different combinations that give rise to that overall oligomeric state. In other words, a 16-mer could be 8 and 8, or it could be 9 and 7, etc. And so with the mass spec, we can quantify the relative abundance of all of those for the particular slice that we interrogated. So what we do after that then is, rather than just interrogating one slice and stopping, is we then do a range of slices and obtain similar mass distributions for each of them. And what we were able to do in this experiment was to interrogate something approaching half of the overall oligomeric distribution. At the higher mass end, so above around about 11,000 mass to charge, it became too difficult to actually do that mass spectrometry experiment. It was no longer feasible to dissociate these larger oligomers, probably because of their more degrees of freedom that they have. So nonetheless, we can reconstitute an overall oligomeric distribution from these colored slices, and that's what's given here at the bottom. So this is our mass distribution, color-coded according to the selections that we made. And if you compare that to the distributions that we have from the original oligomers, clearly there are many more oligomeric states consistent with this heteroligomerization concept. So you can view that then as a sort of a two-dimensional grid where you've got the number of subunits of the first protein and the number of subunits of the second protein, and their combination gives you the oligomeric distribution. So you get this sort of checkerboard-looking effect that actually arises from the fact that oligomers with an even number of subunits of small heterotropic proteins, a total number of small heterotropic protein subunits that's even, are preferred over those with an odd number. So that is as far as you can get with the mass spectrometry on its own, but we really wanted to be able to actually understand quantitatively the biophysics, and we had this problem that we knew we only interrogated a subset, a sub-fraction of the overall mass distribution. So the approach that we took was, first of all, to interrogate our mass distribution, so this grid essentially, using the language of basic chemical kinetics. So we could think about the fact that oligomers could exist in a state with an even number of subunits. They could then gain a subunit with a certain on rate constant, multiplied by the concentration of your small heterotropic protein subunits, to give you an odd-numbered oligomer, so one that contains an odd number of subunits, and that could gain a further one to then become even again. And similarly, in the other direction, you could lose subunits. You could lose subunits from those that have an even number of subunits or those that have an odd. So this is a basic biophysical model that we were considering. And the idea is to use that model together with the mass spectrometry data and mass photometry data to try and understand and extract the rate constants that actually underpin assembly. So this is a rather full slide, but I'm going to try and take you through it in some detail. So the mass distribution I showed you before that we obtained from native mass spectrometry is in purple, and then a mass photometry distribution is in gray in the background. So you can see they line up relatively well, but as anticipated, the mass photometry gives you a full distribution of the molecules in solution, whereas we know our mass spectrometry data was inherently biased towards low mass, because that's where we could select the molecules and do the tandem mass spectrometry experiment to make them fall apart. So we kind of know that the mass spectrometry gives us good sort of micro heterogeneity information, but it doesn't do very well at giving us information over the overall sort of macro heterogeneity. So what we can do is take advantage of this correlation of the two approaches and filter the tandem mass spectrometry data. So filter the purple data set according to its correspondence with the mass photometry. And sort of in rough terms, you can view that as just being sort of a red line cut off here. And translated into the oligomeric distribution, that essentially means that you're truncating part of the overall oligomeric distribution. And we're not going to take this as being real, but we will take what falls within the confines of this red boundary as being an accurate measurement. Then we can take our kinetic model and fit it to the oligomeric distribution within here, and we can consider rate constants associated with the addition and subtraction of each type of subunits, and that gives us an overall distribution then of the oligomeric states that are present in solution, which is this whole large checkerboard now, right? So that's what we predict based on a kinetic model. And then we can say, well, does that model correct, or does that seem reasonable? And we do that by reconstructing the mass distribution that this grid gives us, which is now this light blue mass distribution here. And you can see, again, that overlays pretty well with the mass photometry. So in other words, we've used the mass photometry to leverage a larger space of oligomeric states that allows the kinetic model to actually extract values that are most consistent with the oligomerization of this protein. Now, what I will mention at this stage is that, interestingly enough, we found that the on and off rates for these two proteins did not require them to be any different from each other. In other words, one set of on and off rates was completely sufficient to reconstruct the oligomeric distribution. So these are two different proteins, and yet they behave as though they're not in this type of analysis and this type of experiment. So I'd like to just finish and sort of describe a little bit to you, if we, the idea of a sort of continuum of protein assembly. What I've showed you here is a situation where two proteins, so I would indicate that with number of subunits of I, number of subunits of J, have co-assembled to form a distribution of oligomers, and one that's essentially just what you might expect statistically. In other words, it's governed by the number of possible combinations of which one can make certain states. And that's manifested by the fact that the rate constants for on and off, for coming on and off those oligomers, are identical for the two proteins. And it's an interesting point just to consider from a biological point of view that those two proteins are separated by several 100 million years of evolution, and yet they have neither evolved to specifically self-assemble or specifically co-assemble. And there's a lot of science there that we still don't understand. So just as a way of summary, I hope I've shown you a little bit about how we can use mass measurement approaches in biophysics and structural biology, namely native mass spec and mass photometry and their combination to understand the structure, thermodynamics, and ultimately kinetics of protein assemblies. And I'd like to just finish up by highlighting the people in my group currently and previously whose work I had the pleasure of showing today, in particular Miranda Collier and Dominic Shaman for the project on heteroligomerization I described. I'm obviously very grateful to long-standing collaborations with colleagues in Oxford and collaborators outside, and my various sources of funding. And thank you very much for bearing with us.
Thank you, Justin. That was very interesting. And now we will move on to our next speaker, Nikolas Hundt.
Thanks very much for this invitation to the webinar. I'm very excited that I can share some of my insight into this relatively new technique called mass photometry, which is based on interferometric scattering microscopy. When I was a postdoc in Philip Kokura's lab, I was interested in this protein called actin, which polymerizes into filaments, as you can see in this image. And the lab had previously shown that it is possible with iSCAT to image individual proteins entirely label-free. So I was interested when I joined what new details we could reveal with this new technique on the actin polymerization process. And what I've seen, I want to share with you in this webinar. Actin is a major component of the cytoskeleton, and it can be found throughout the cytosol, where it forms all kinds of different structures that give the cell shape and help its movement. As you can see here, for example, you can find it as filaments at the leading edge of a migrating cell, where it protrudes the membrane forward with this lamellipodial network, or you can find it in structures that are called stress fibers, which the cell uses to retract its rear during migration or to attach itself to the extracellular environment via focal adhesion. And if you label actin with a fluorescent marker inside cells, it can be used as a very nice tool to visualize all of these structures live in cells in a fluorescence microscope. This way you can very nicely study the architecture and behavior of the actin cytoskeleton. However, if you want to understand the molecular mechanisms of how these structures are formed and how different actin binding partners interact to regulate their formation, you need to understand the fundamental mechanism of how actin polymerizes into filaments. And the best way of doing this is by isolating the protein from cells and investigating its intrinsic properties in a controlled in vitro environment. Actin is a fairly well-studied protein. The first studies have been around in the 1960s, and that also makes it a nice candidate of explaining what the actual experimental difficulties are if you want to study the assembly of a biopolymer. Actin itself is a fairly globular protein, as you can see here, which polymerizes into these double-stranded helical filaments. And it does so via a nucleated growth mechanism, which means that a couple of these monomers have to come together to form a stable intermediate, which is called nucleus. And from that nucleus onwards, the actin filaments can elongate very freely. We know the structure of these filaments fairly well from imaging techniques like electron microscopy, or more recently also from atomic force microscopy. And these give very detailed insight into the structure of these filaments. For example, in these AFM images, you can directly see the helical nature of these filaments. And more recently with high-speed AFM, it's even possible to record these things dynamically. Even more recently, it has also been possible to generate an atomic model of an actin filament using cryo-electron microscopy. And earlier it was already known how the atomic structure looks of a monomeric actin from X-ray crystallography studies. That means we know fairly well how these proteins look. However, if you want to study the assembly of such a filament dynamically, it becomes a lot more difficult because you lose a lot of information because you can't use these high resolution techniques that I've mentioned before. The usual way of doing this today is usually with fluorescence microscopy, where you label part of the actin subunits fluorescently and then watch the growth of individual filaments in the microscope, as you can see in this time series here. You can somewhat extract kinetic parameters from this experiment. For example, the elongation speed of the actin filament from tracking the tip of a filament. And this technique is also very useful if you want to understand the influence of actin binding partners on this process. One example in this video shows how a mix of actin with a binding partner called ARP23, which is an actin filament side binding protein. And this binding partner binds to the side of an actin filament and branches of a new filament, and thereby forms these dense actin networks, similar to the ones that are observed at the leading edge of a migrating cell. And from this video, you immediately see how this binding partner actually works. If it comes to the nucleation phase, and if you want to study that, it becomes a lot more tricky, because the techniques that I've shown you before don't really have the sensitivity to detect these tiny species that are present at the very beginning of this reaction. And what people have done so far to investigate this process is by looking at the bulk signal of a polymerizing actin solution, where you basically follow the overall bulk signal of how many filaments there are in the actin solution. And this can be done by different means, for example, fluorescence or light scattering, or you can measure the viscosity of your solution, or by birefringence. And in all of these cases, you get a signal and time course that looks like this, like a sigmoidal curve, where in the beginning you have a nucleation phase where nothing much happens. And then as an appropriate amount of nuclei have been formed, the reaction speeds up and you get more filaments up until a point where the filament concentration reaches a point where the depolymerization and polymerization rate become equal, and then you reach a steady state where you don't get any net growth. Now, from these curves, since you get some information about the nucleation in the lag phase and some about the polymerization phase in this region of the curve, you can extract kinetic parameters like a nucleation or polymerization rate. However, since these techniques are basically blind towards the individual molecular species that are involved in this process, you get somewhat averaged rate constants for these processes. But you don't really know what the species actually are that are involved in the molecular details. That means up until today, it's not really possible to extract the microscopic kinetic rate constants between these individual species, and you don't really know experimentally what species there are that do nucleate actin filaments. The same is true if you were to ask the question, what species actually attach to a growing actin filament while it's elongating? Because in all of these cases, what you would need is a method that quantifies all the species that are present at single subunit sensitivity, and to image the whole process in a dynamic fashion. The next question that you could ask would be, what method could provide all of these requirements? And you might have guessed it, the short answer to this question is interferometric scattering microscopy and mass photometry. Philip and Justin have already explained this quite in detail, but I think it doesn't harm if I just briefly go through the basic principle of it again. In iSCAT microscopy, you shine a light onto your sample, which usually consists of a glass cover slip and an aqueous solution on top, which normally contains your protein of interest. And then you collect the light that comes back from your sample and separate it onto a camera. And the light that comes back from your sample is a combination of the reflected light from the water-glass interface and scattered light from small objects that sit on that interface. And if you go to a position that we define as focus, these two light fields interfere destructively with each other, such that these small scattering objects appear as dark spots on a bright background. And the advantage of this approach is that the interferometric scattering signal, which is represented by this third term here, where R designates the reflectivity of a point and S the scattering amplitude, you can see that the scattering amplitude or the interferometric signal scales only linearly with the scattering amplitude, and therefore for very tiny objects this whole signal disappears much slower than the pure scattering term. And that way it is much more feasible to detect very tiny objects. We have been able to this way detect individual proteins entirely label-free. If you now put a drop of a protein onto your glass cover slip and just record a video of what's going on, you will get a movie that looks approximately like that, where each of these dots that appears represents a single protein that lands on the glass surface. The reason they disappear again is because of the way we process the videos, where we constantly remove the static background that is generated by the glass surface. And as a protein lands, it over time becomes part of the background and is therefore removed as well and disappears from the video. Now you may notice that some of these dots are actually fainter and some of them are darker. This is because the objects that we are observing and their signal scales directly with the size of the object. And for proteins, this is very convenient because all proteins have a fairly similar refractive index and a very similar specific volume, and therefore the scattering signal is directly proportional to the mass of a protein. And if you now take a set of proteins where you know your mass and measure their scattering contrast, you can use this to calibrate your scattering signal and convert it into a mass signal. That means if we now determine the mass of each of the dots in the video that I've shown you before and plot these masses in a histogram, we will get something that looks like a mass spectrum, and each of the species that is present in your solution will give you an individual peak. Now, this particular video that I've shown you before was a solution of actin shortly after the initiation of its polymerization reaction. And if you apply exactly this approach to such a video, you will get a size distribution that looks like this, where you have in the beginning a peak that corresponds to actin monomers, which is 42 kilodalton. But you also see these smaller humps over here, which are at equally spaced mass distances to each other. You see a zoom in this picture here. And this is very easy to interpret because these are basically just the individual oligomeric species of the actin. And this is really powerful because now in a very narrow time window, we can just very quickly record the size distribution of our actin solution. And as we can do this at different time points after the polymerization start, we can directly follow the nucleation process of actin in real time with single subunit sensitivity. And we can not only do this at different time points, but also at different actin concentrations. And this way we get a global overview over the behavior of the system. And here you can, for example, see that you need to actually increase the actin concentration beyond a certain critical one, which is similar to the one that's been reported in the literature before. And only then you get eventual net growth of your distribution after later time points. And since we can follow the individual concentrations of each of the individual species under all these conditions, we can develop a kinetic microscopic model of the entire system. And we are currently in the process of doing that. And in our first attempt, we have assumed a model where, for structural considerations, we have just assumed that the first two steps have different equilibrium constants than all the subsequent steps. And now we can take the information that we have from our experimental distribution and determine the relative abundances of all these species at the start of the reaction, feed it into our simulation, and then run a simulated reaction and see whether our simulation, here designated by the red curves, arrives at our experimental distribution at later time points. Now, the goal of this would be to have one set of rate constants and which explain the data of all our actin concentrations at all the different time points. As you can see, we are not quite there yet. There are deviations still, but we are approaching this goal to a certain extent. Earlier, I've also mentioned that it would be interesting from a mechanistic point of view to investigate what species actually attach to a growing actin filament during its elongation. And during my postdoc, I was interested in whether this is actually feasible with iSCAT microscopy too. And for this, I have applied an assay where I attach partially biotinylated actin filaments to an avidin-coated glass surface, and the surrounding solution contained actin subunits above the critical concentration for filament growth, and therefore these free actin filaments would elongate these free subunits would elongate the actin filaments on the surface, and I could directly follow this process live in iSCAT. As you can see here with these dark growing filaments. And since each of the species that would attach to an actin filament gives me the exact same signal, and I don't have effects like photo bleaching or blinking as you would have it in fluorescence microscopy, I could follow this process at very high precision and relatively high frame rate. And if I would now just track the filament tips as they grow and determine the displacement of these filament tips over time, I noticed that there are incremental changes in the length of the actin filament. And the step sizes of these would be approximately the size that you would expect for the attachment of individual subunits. And if I would now take the frames that correspond to such a plateau and subtract the frames that correspond to the previous plateau, I would get a different image that gives me a scattering signature of whatever protein species attaches during that time point. Here you can see the shadows or the scattering signatures for different step sizes, and the larger the step size, the darker these signatures are, of course. But since I have my mass calibration, I can now go back and ask the question, what mass does this shadow actually correspond to as the filament tip grows? And in fact, if you do that, you'll see that a step size of 2.7 nanometers, which you would expect for a single subunit addition, corresponds to a mass of approximately what you would expect for a single actin subunit. And for larger step sizes, the scales correspondingly. You can now do this for a lot of these steps and generate a size distribution of what is attaching to your actin filament tip. And as you notice here, there are not only step sizes that correspond to single additions, but also to double and triple additions. some of which are probably because of our limited time resolution. You will also record 2 consecutive attachment events within a single frame. But to a certain extent, this also shows that there are dimers and trimers attaching to the actin filament tip, which is feasible if you consider that there are dimers and trimers present in solution, as we've seen in the nucleation experiments before. Now, bare actin, of course, is fairly boring if the goal is to understand how actin polymerization works in cells, and you would like to look at how this process is actually regulated by actin binding proteins. But the basic assay systems that we've developed for bare actin can directly be applied and used to investigate the mechanism by which actin binding partners influence this process. The most straightforward experiments would, of course, be to just look at the actin elongation in the presence of an elongation factor, such as formin here, or in the presence of a depolymerization factor, such as cofilin. Another interesting question will also be to ask, what is the nucleus size in the presence of a nucleating factor, which again, formin also nucleates actin filaments. And we're in the process of also asking and answering these questions. Now, going beyond actin, there are also other biopolymers that would be that one could study with the exact same set of methodologies. And one prominent set of biopolymers, which basically suffers from the same lack of methodologies, is amyloid fibrils, which are involved in the pathogenesis of a lot of prominent neurodegenerative disorders such as Alzheimer's and Parkinson's disease. And here it would be extremely helpful if one had a technique that is able to characterize the early nucleation steps, because these early nucleating oligomers are a valuable and preferred target for developing drugs that prevent the formation of these amyloid fibrils that cause the diseases. At the end, I would like to thank a number of people, in particular these people from the Kukura lab, who built the microscopes and helped me with parts of the data analysis. I would like to thank our collaborators who provided me with proteins and advice on data analysis. And I would also like to thank Claudia Feigl for giving me a new home in Munich and for supporting me in starting an entirely new iSCAD project. And thank you very much for your attention and for attending this webinar.
Thank you, Justin and Nikolas. That was an excellent presentation from both of you. We have a few questions from the audience. If anyone else has a question in the meantime, please feel free to post that in the questions box that appears on the right-hand side of your screen. So to start you out, Justin, what are the main advantages of mass photometry compared to other mass measurement approaches?
Of course, I think that question depends a little bit on what sort of comparisons you're trying to draw, which methods you're comparing between. But I think for me, what really stands out is really the ease and the speed of the measurement. It really is a matter of just a couple of minutes to obtain a mass distribution measurement. And that really compares very favorable to all other methods that at least I'm aware of out there. It's of course incredibly attractive. The measurement is in solution. That's fundamentally relevant to pretty much all biology, I would say. Despite being in solution and the complexities that causes, it gives you really quite high resolution of separation. In other words, the mass distribution you measure is well resolved in comparison to other methods, I would say.
Mm-hmm. And there's another more specific question that came in during your talk. For hetero-oligomerization experiments, like the one that you showed for HSPB1 and HSPB5, is there a requirement on the range of dissociation constant of the heterooligomer for mass photometry to work?
I mean, I think that's a sort of a general question in a sense. I don't think there are particular restrictions made when considering heteroligomerization, in the sense that the method will detect whatever oligomers are there. It's independent of what the mode of oligomerization is. It just measures mass. Of course, you know, like any method, you have to consider the concentration you're making a measurement at relative to the association or dissociation equilibrium constant of the protein and protein complex that you're thinking about or that you're analyzing. But I don't think there are any specific restrictions otherwise. So of course, you know, you can perform the experiment at a range of concentrations, and that's something that is valuable when you're trying to probe an equilibrium. And actually, you know, you can even use that to your advantage to quantify the equilibrium constant by assessing the relative abundance of associated and dissociated species in the measurement.
Great, thank you. I've got a few more questions. Well, we have 1 or 2 minutes left. Nikolas, could mass photometry be applied to a complex mix of biomolecules?
Well, thank you for that question. I think it can be applied to a complex mix. I mean, you've already seen from Justin's presentation, for example, if you mix 2 molecules that form quite a wide range of different species, you can separate the different species very well in the mass distribution. And the same, I would say, is true if you were to mix a lot of different components. Of course, you need to consider, first of all, the best would be if you know the mass of all your components that you put into your mix, because then it is easier to assign the peaks that you observe. And then it also comes down to how similar in mass these different components are. For example, if you were to try and separate 2 components with very similar mass, the peaks would probably fall on top of each other, and our lower resolution limit is somewhere in the range of 10 kilodalton, 10 to 15 kilodalton.
And kind of related to that, what would be the smallest protein that you could reliably detect using this method?
Yeah, that depends on a couple of factors, of course. I would say actin is already quite a small protein to detect. We have gone down to like 30 kilodalton proteins, but the problem at this low mass regime is that you run into a lot of trouble with the noise that you might be detecting as well. Yeah, so I wouldn't recommend going below 30 kilodalton, definitely.
Well, we have time for one more question. We do have several more questions, but I can guarantee the audience that the speakers will get to see all the questions, but we only have time to include one more. Can we only measure molecular weight, or is it also possible to measure the size of the proteins with this technique?
I can answer that, Ava, if helpful. So the mass photometry method really is mass sensitive. You know, that's what you quantify. So within the diffraction limited nature of the methodology, we have no evidence that we see differences based on structure for isobaric, i.e. molecules of the same mass. So what you really have here is a pure mass measurement approach. And that's how best to think about it.
Mm-hmm. Well, great. Well, thank you very much. And that brings us to the end of this seminar. So thanks again to Justin Benesch and Nikolas Hundt for a very illuminating presentation and a great discussion afterwards. And thanks also to our sponsor, Refine. And finally, thanks to you, the audience, for taking the time to attend and listen in. If you have enjoyed the seminar and would like to view the video recording of this session, please visit the seminars page on bitesizebio.com, and it should be available within the next 24 hours. And there you can also see the other webinars we have lined up for you on Bitesize Bio. So until next time, good luck in your research, and goodbye from all of us at Refine and Bitesize Bio.